I've had a few questions on lab 1 with the graphing. Now, the reason I think there's some confusion with the graphing is that you're given this sort of general table and given some instructions, very, very vague instructions on how to graph. They don't actually tell you what you should be graphing. And the reason is that this lab, as I said in class, was adapted from another class so that we could use it for our first lab instead of the lab that we were told we couldn't do because of COVID. So in the original lab that we would have done for this class, students are asked to design an experiment in a different way, and the instructions are left to be sort of vague because there are a variety of different assignments that you could possibly do as a class. So this fish lab is a little bit different than we have been doing, and again, it is also vague because each class might choose a different type of experiment to do. But based on the experiment we did in our class, you are actually going to make a line graph. Now, if you recall, or when you watch our lectures, there are different reasons that you make different types of graphs. You make a line graph when you are looking for trends or making comparisons. Okay, the other one that students like to try to do on this graph is a bar graph, and it just doesn't really make sense for the type of data that we have. So while we were in class, we talked about the independent variable. So I'm going to use this opportunity to just sort of review what an independent variable is. An independent variable is the variable that you are testing or controlling. So it's the treatment that you change. So this is the treatment. And then the dependent variable is the data that you collect. Now, it's called the dependent variable because it depends on the treatment that is happening in the independent variable. So our graph here asks us to label our axes appropriately. Your x-axis is independent and your y-axis is dependent. So in our case, the x-axis is going to be our independent variable, which is our temperature. Now, each group used a slightly different temperature, but they were all written as a range. And so I'm going to pause here and label everything. Before I do that, I'll label the y-axis at the same time. On our y-axis, we're going to label our dependent variable. In this case, our dependent variable was the number of opercular movements per minute. Now, I gave you a set of data because we ran out of time with talking about how the class is going to run and all that. So I used a data set from a previous class that did an almost identical experiment after you guys did your experiment real briefly, so you understood the process. And then within that data, you were to calculate an average. So there's an average column, and then there is a column for each of the fish that you guys did. Now, since I gave you data, you will just use that data. When you make your data, when you make your graph, you should draw your fish, a line for your fish and for the average. Now, I didn't mention that in class. We ran out of time. So you would want to draw your fish and an average line. And then you're going to use that information to sort of determine if we have supported our hypothesis that the opercular movements and therefore the respiratory rate increases with temperature. So I have labeled my axes, and with the values that we need. There's not a specific right or wrong way to do this, in the sense that you could number your y-axis by 5s, you could number it by 20s, you could spread them out a bit. The only thing you need to keep in mind here is that you need to keep all of the data on your graph. So I just did this quickly. I didn't look at all my data. You need to do that though. On the bottom, I have labeled our time frame. So I'm going to graph for you fish number 1 on our data set that I gave you. So for 7 to 9 degrees, we've got 69 opercular movements, followed by 68. And I'm going to jump ahead here. And just to move things along, I'm going to finish off my graph here. So this is my line for fish number 1 from the data set that I gave you. We'll go ahead and connect this. You should also draw the average that you calculate for your data. So you would have also written down your data for your fish. You can go ahead and add that in. We did not collect a whole range with one single fish, so that's why I'm asking you to use the rest of this data. But now you would draw, this would be fish number 1. The graph also asks you to do a key, so you might say blue is fish 1. And then, let me change my color here. Oops, that's not the right color. And then we might have our average with red, and that average might be something like this. I don't know what it's going to be. I haven't graphed that. But you'll use that information to state your conclusion. Did you support or did you not support your hypothesis? And then you can continue on with your application questions.